Okay, I got this message today, and it kind of messed up my schedule. So I guess we're not going to have the meeting on Monday. And there's a good chance that once we get into the early part of July, starting with the first set of the meetings in July, they may be longer. Okay. It's a good chance I would need to start those meetings at 5 o'clock.
I'm sorry, Professor, could you repeat what you just said?
You see this message just sent me, district announcement?
Yes.
Okay. Did they send this message to you also?
Yes. Did you say something about meeting at 5 o'clock?
Most likely we'll be meeting at 5 o'clock starting in July.
Every day?
I need to catch up with the, I need to make very certain that all the topics are taken, have gone through for lecture exam 2. In most of the past semesters, students tend to think, well, most of them would say that exam 2 was the hardest.
Oh my God.
Simply because exam 2 has 4 body systems. Okay. So I want to make very certain that I get those through before the exam. So you want to make very certain that you have some more time to go over that material before the exam.
So on Tuesday, we'll start at 5 and that will continue till the end of the course or just for the week?
We'll see. We'll see what happens on Tuesday. Tuesday, I'm not changing those those schedules. But once I post the July ones on Canvas, yeah, most likely the first week or 2 weeks of of the July ones would be starting at 5. Okay. So when I send you your scores for the exam, you will see the ones that you miss. And then you can ask what those answers were the next meeting. So the next meeting would be Tuesday.
Professor, can I ask you a question?
Yeah.
How many people drop the class after the first exam?
In past semesters, usually the ones who don't do very well. And this is the first time I had over 10 people leaving the class before the exam. Okay.
Professor Chinn, do you give extra credit just to help boost our grade?
Yeah, I'll be going over that when I send out the next message, probably. Has to do.
Give
Yeah.
extra credit for what? Can you repeat that? Sorry, I had a little
It
break
has,
up in my.
it has to do with two, well, anywhere between half a point to 2 points of your grand average of the semester. Has to do with doing a research presentation, and best way to deal with that is to do the research presentation do it on YouTube, so the rest of the students can get to it. Let's see.
How many points would that be worth?
2 semester points. Has to do with when somebody is right at the border between 2 grades, that can readily get them to the grade higher.
Is that like 2 points as in 2% of our whole grade?
Your grand average
Okay, thank you.
of the semester.
And how would that be graded?
Let's see. This one. Okay, yeah, these are the ones. These are the ones for Kenyatta. I give 3 points at College Marin because that's a 5-unit course. Okay, so I would tell the entire class to go through that, and they can do the scoring anywhere between half a point to 2 points. Okay, and then I'll get the average and then I'll add it to that person's so the way you can do the research presentation, you can do it by yourself, or you can have 3 people together doing it, just one topic. And the only thing is that for the presentation, we need to hear different explanations from each person. Okay, we don't want to be listening to the whole same thing over and over. Okay. And yeah, this is what happens. I need students to send me their their scores and a short a short critique. Okay. So, and I'll start showing some videos after the second lecture exam, and that's kind of like my examples of things that you should go after. The type of topics has to do with something that you can readily explain with anatomy, other than giving us all kinds of terms and items that nobody can understand. Okay, like molecules and chemicals, etc. Okay.
And
Yep.
Professor, what
Yeah.
percentage of your class would you say is covered in pathophysiology?
Not by much.
But it'll prepare us for that class, right?
Yeah. The reason I say it's not what I've gone through with the functions of items in the body. In past semesters, there would be people that told me that most of that is already covered in their physiology course, but that depends on who's teaching those courses. The reason I'm doing this is because in most of the 4-year institutions, physiology after anatomy is usually you need to go through with arithmetic. You need to actually go
Okay, thank you, Professor.
yeah, you need to actually go through all parts of a nephron in the kidney. About the concentration of material. Okay. All right. Many of the sensory receptors in the integumentary system would be showing up in the nervous system chapter. So you would find these in chapter 14, but the set that they have in the on the table here are essentially items that would be in the skin. So, and there are two sets of receptors. You have one set that would be encapsulated, and the set that we're looking at now, these are the unencapsulated ones. By that, we are saying that there's no items forming around the branch of the sensory neuron. So largely, it's almost like a bare branch of that sensory neuron. So free nerve endings are the ones that would be picking up on light touch or a little bit of pressure. Okay. And so in your textbook table, we go over these functions. And mostly for the set of material here for the free nerve endings, they are the ones that's going to be picking up on temperature. And also they'll also be picking up a little bit of the pressure at the skin. And also some chemoreceptors. And the major function is essentially nociception. Nociception is picking up pain. So the free nerve endings in the skin entirely microscopic, and those free nerve endings is pretty much like a sheath of these free nerve endings, very close to the epidermis, and some of these items, often referred to as dendrites, would have gone into areas between some of the cells, living cells, in the epidermis. So, yeah, so epidermis. And why did I say that? For the main function nociception, has to do with the excess exposure to temperature. When something is very hot, as well as something that's very cold, these are the ones that's going to be taking in and sending the pain sensation to you. Same with mechanoreceptors. Any heavy pressure that happens is going to be painful. Any chemicals that is entering your skin, that can also be painful. So that's what I mean by for nociceptors. And because the nociceptors is a whole series sitting along that part so next time when you are trained to do the vaccination on people, that needle get through as quick as possible. Unless you don't like that person, then you keep it somewhere around there. Okay. Then this next one is the epitheliotactile complex, used to be referred to as the Merkel it's not just the Merkel disc. Down here is that disc, but has to do with these two items. The cell, the tactile epithelial cell, is the one that would be sending chemicals onto this disc-like item here. This also would be the part of the dendrite of the sensory neuron. And So any kind of light pressure. What do we mean by light pressure? And why is it that you're picking up the sensation of what you are touching or what is next to your skin? In a living subject, you have blood vessels here, capillaries, as well as all kinds of larger blood vessels a little bit deeper. So you constantly, even though you may not sense it, you constantly would have pulses here. And that pulse is the one that's going to be constantly moving all parts of your skin, even though you're not picking that up. But whatever is resting on that on your skin, you would immediately know. In other words, right now, you're you may not be fully figuring it out, but immediately at any one point, you know what is on your skin, the type of clothing, whether it is cotton, silk. Okay. So similarly, this is also the one that allows you to immediately feel whenever you put your hand or any part of the skin of your body on the surface of, say, the table, floor, or whatever, and immediately you can figure out whether it is glass, wood, stone. Okay. So that's what happens with these ones that are way up in the superficial part of your skin. And then there's this one. You have some of these free nerve endings that come to the hair follicle. And supposedly this is the one that's going to be telling you that which way that hair is going to be moving. Okay. Or sometimes have a small insect is walking around here kicking over a hair. This is the one that's going to be giving you the sensation of it. But what is done here isn't exactly accurate. You actually have more than one set of these sensory neurons here. There is another one on that side, okay, on the other side. And that has to do with most of your most of the other mammals that would have fur. If you stroke the fur in one direction, in the direction where they usually would be lying down, and that animal would feel comfortable with that. If you do it the other way, that animal would not like it. Okay. And that's where that statement came from. When people say, rubbing me the wrong way, okay, has to do with pushing the hair going the other way. And it's a very different sensation. So these are the encapsulated items. Then
Professor, really quickly, when you're looking down that center column, like exteroceptors, interoceptors, and proprioceptors, extero, that is kind of talking about like external, internal, and proprioceptors, what are those? Up top under free nerve endings.
proprioceptors are the ones that would be picking up on the change of the shape, whether something is stretching. Yeah, proprioceptors are the ones that's going to be picking that up. Okay. And definitely you would have quite a bit of proprioceptors at the joint. All the synovial joints would have that. So any kind of stretching or recoiling is going to be picked up by that. So these are the encapsulated ones. In other words, there's going to be a capsule. And then you have the tactile corpuscles. These are the ones that are up at the dermopapillae, the dermopapillae. Okay. And these are the ones that's also going to be picking up light pressure, as well as vibration. Well, I just stay with pressure. Okay. And the reason I just don't go with vibration, because vibration simply has to do with pressure, release pressure. Okay. It's a problem here that when they generalize the the actions of those parts of the body that would have something like this. Okay. A few years ago, researchers checked this out. just because you have corpuscles that are somewhat similar, and they can all be regarded as tactile corpuscles. But in different part of the body, they actually give different they actually send in different sensations. Okay. And those sensations very often would be going to different parts of your brain to pick those up. And what's the term for the material that is sensed with the fingertips for the blind people?
Braille?
Thank you.
Braille?
Yeah, Braille. The researchers a few years ago, the people who can readily figure out Braille with their fingertips, and they get them to use their genitalia on that material, and none of them could figure out what they are touching. Okay, so now we can readily figure out that the structures there have very different would pick up very different senses, and they would be sending the material to, simply because they're sending it to different parts of the body, and different parts of the brain, then you can always figure out what's happening with it. Handling those sets of tactile corpuscles. So, yeah, can't do Braille with your genitals. So don't try that at the ATM. Lamellar corpuscles, these are the ones that are relatively much deeper, and some of them would be in the hypodermis. Okay, both dermis and hypodermis would have these. So deep pressure is going to be picked up by all these layers in that capsule. The bulbous corpuscle, this is the one that largely would be doing proprioception. Okay. So picking up on any kind of deep pressure, stretch, etc. Okay. So also relatively deep in your dermis, hypodermis, and all the joint capsules would have that. And So epidermal appendages, we're going through hair and nails. So once the heart stops in a person, blood will no longer be circulating, and oxygen would not be coming to these cells. So your hair and nails do not grow after death. Okay. Sometimes people do those measurements and they claim that they get longer. It has to do with after a person dies, the skin may start shrinking, drying up, and when they do the measure, when they do the measurement at that time, they would think that it got longer, but it never did. Okay, it's just that the skin shrink back from it. So we'll start with the hair first. Hair is the product of the epidermis, and that means a good portion of the material in your hair would be from ectoderm. So now you can see that all those stratified squamous cells infatuated to form that hair follicle. And then at the very bottom of the follicle, you have this part that is referred as the bulb. Way at the bottom of the bulb, you have this item that is referred as a hair papilla. Hair papilla is not from ectoderm. Hair papilla would be from mesoderm. Okay, so this is what happens when you have blood vessels and nerves to be brought in by the mesenchyme. So when that pushed in here to form that hair papilla, then this set of material that had invaginated in here would now be reflected around that papilla. And the items here would continue to differentiate into doing something else. So now what you are seeing is some of these labeled items. You have to fully understand the part that they put down as glassy membrane. Mostly because in a living subject, this item may not have any color in it. So that's the reason they use that term here. If you ever pluck a hair out and you see something that is transparent around it, colorless. That's not the basement membrane. Okay, that's not that. It's not that item here, which is the basement membrane that is labeled as glassy. If you pull your hair out and you manage to rip this basement membrane, you will get blood coming out. Okay. The piece that you actually see that is transparent, glassy, is this one, the cuticle. Okay. So that's a differentiate. That's when the actual part of that hair shaft is wrapped around. So that hair shaft would be wrapped around by that. And again, those are cells that would pretty much start dying once they get up to the part that is referred as a hair root. Okay. So now at the basement membrane of the hair papilla, you would have cells that have somewhat differentiated, and you get melanocytes here. Those are the ones that's responsible for your hair color. And then you have this set of cells here that would be constantly mitotically divide and send the daughter cells out. Hair matrix. The matrix is the part that is responsible for the elongation of the hair. Okay. Not the ones out here. Just here. And all this is coming from it and continue getting pushed further and further up. Once you get past the hair root, they will all start dying. So different types of hair in a person. Lanugo is the hair that had developed and grew in the on the fetus. And that's why when some babies are brought out on their back, you have those sets of very long hair on their back. Okay, and usually within a week, they would all drop away. The phallus hair, sometimes referred as a down hair, would be those very thin somewhat short hairs throughout your body. Okay. And then you have the definitive. These days, many textbooks would use these two terms together. When I took anatomy, these two terms actually had to do with different types of hair. Okay. But now they're used interchangeably. But actually, definitive hair would be let's see. Make sure I have this correct. Yeah. The definitive hair are the ones that only come to a certain length and do not continue to elongate. Okay. So has to do with, say, your eyelash and the ones in the eyebrow. So those would be the definitive ones. The terminal ones would be the ones that continue to lengthen. And usually those ones, both of those two would be, would start dying between after 2 to 5 years. So, and that's the reason people who never cut their hair, the hair would only continue to maybe about a little bit over 4 feet. Okay, but mostly somewhere around 3 feet to 4 feet. Yeah, the main thing I need you to pick up on here is the matrix. Okay, matrix is the one that is responsible for lengthening the hair. Sebaceous glands are the ones that associate with hair follicles. So only those parts of the body where you have hair follicles would have oil essentially wrapping around that the hair as the hair continue to push out. The rectus pili muscle, say smooth muscle here, and because it's a smooth muscle, you are not able to work on it. It's not like your skeletal muscle. So this one, the cells in here forming that rectus pili muscle would respond to temperature as well as some hormones. Okay. When the temperature drops, ambient temperature starts dropping, then they would start to contract, and that's when you get those goosebumps showing up. Okay. There are other times that you also would have goosebumps. And that has to do with the part of the nervous system that's referred to as the ANS. So ANS, autonomic nervous system, is the one that is responsible for having a subdivision that is called the sympathetic division. And the sympathetic division is the one that respond to emergency situations. So if you're walking at dusk and something jumps out in front of you from the shadow, immediately that autonomic nervous system is going to be getting the erector pili muscles to start working. Same thing with the other mammals, the one with fur, when they are scared, the erector pili muscle would draw up all the hair, make them look bigger. Okay. But with us, we don't have enough hair to show that. But this will also give you a series of goosebumps. Both hair and nails would be hard keratin. Okay. And that's how tough the keratin protein can be. So once the hair start to continue out after they've been shaved, you get that sensation because it picked up the cutting edge of the shave of the shaver. Okay. Someone must have counted these. So yeah, most people would have that many hairs in the scalp, and each day would lose that many. each follicle is capable of producing 30 feet in a lifetime. Okay. But if a hair is never cut, it would get to about 3.5 feet long. That's when that follicle needs to be replaced by a new one. The shape of the follicle is the one that's going to be determining whether you have straight hair or curled hair. So all the pictures shown in your book would be straight hair follicles. Okay. If the ones are coiled, then whatever comes out is going to be curly, largely because you're dealing with hard keratin. So it will stay curly. Yeah, so both hair and nails. Hard keratin. If the melanocytes are the ones that would be drawing in some iron molecules, then the hair would look a little bit reddish. All the other colors would be the result of the black melanin. And as you age, at some point, the melanocytes would not be sending out as much of the melanin granules. And especially if some of the cells take away the granules that would be there. And of course, shortly after that, those cells will be dying. And without melanin, you now would just have air in that hair shaft. And that's the reason it's going to show up whitish or silvery. And it's very much like in the in the fishbowl. And if you have an air stone in it, and you see that whole set of air bubbles coming up, that's essentially what's happening with gray hair. There are air bubbles lodged in it instead of the melanin granules. Okay. So brunette has more hair than, blondes have more hair. Okay. Now you know when's the best time to shave. Hair grows slowest at night. Start speeding up in the morning. So Okay. Yeah, people can have long beards. So for the nail, this is the hardest type of the so again, when you're looking at the nail, you have this item that is the nail bed, and the part that is referred to as the matrix, that part of the hair is also where the nail is elongating. So again, by the time those keratinocytes get to the the root of the nail, then everything here would be just dead cells. And on the top of the nail, the reason you can see this item that is called the nanuli, it has to do with the healthy part of the person and a person who's well fed. That nail bed, this portion of the nail bed would be a little bit thicker, and you don't see the color of the blood under it, so it's much lighter. When you look at your nails and you don't see don't see that, then there's a good chance that you're undergoing malnutrition. Okay. Your book gives you the term here, the eponychium. Then they should also give you the hyponychium. So eponychium is here. Hyponychium is actually under the free edge of the nail. Yeah, right there. So that takes care of integumentary system. Still one more thing, a couple of more things here. Glands of the integument. The sebaceous glands, the ones at the hair follicles, those are the holocrine type of gland cells. So in other words, once the cell is filled up with vesicles filled with oil, then it will leave. And then somewhere at the duct is when it would be lysed, and all those would be broken, and the oil would now be covering your hair, as well as a good portion be filling up onto the surface. Sudoriferous hair, sudoriferous glands, those are the sweat glands. And you have the merocrine type and the ones through on your body that are referred to as eccrine gland cells. Those should be the merocrine type of sweat glands. Those are the cells that stay with the stay at the basement membrane and just continue to send out your sweat. The apocrine type of sweat glands are the ones in the armpit. The mammary glands, these ones here are also modified apocrine sweat glands. Okay, and these cells are the ones that will be producing milk once it gets the hormone coming from the hypophysis cerebri or the pituitary gland. Ceruminous gland, when you look in the dictionary for cerumen, it's going to say wax, but cerumen is actually not wax. Okay, you look up cerumen, you find at least a couple of dozen chemicals there. So ceruminous glands are the ones in the ear, external ear, and they are modified apocrine type of sweat glands. So those are the ones that constantly send out that material that most likely would have a high level of sodium. So that's why it's bitter. And that's the material that would not allow insects to enter the ear. Okay. So, okay. We start with the biology and muscular system in 10 minutes.
Are you guys still alive?
Kind of tired. Like my eyes are closing.
Yeah, I know what you mean.
You know, it's bad when you're more tired than hungry. Or I should say, so tired that you can't even make food.
Yep. That's what DoorDash is for.
I wouldn't even be able to stay up for it to get here. That's how bad. I could just take a nap, I guess, but
Yeah.
I don't know if I'm gonna make it.
Did you pull an all-nighter last night?
Well, the night before, I probably got like 2 hours of sleep. But last night, I got a little bit more. But I don't know why. I just feel like I used a lot of energy today, even though I didn't really do much.
Mentally drained.
Maybe it's mental energy. Sometimes that takes more than just the regular everyday energy.
Maybe you got a nice bottle of mezcal at your house. Take the edge off.
All I need is my bed and pillow.
Okay. Some of the major characteristics of muscle tissue. Muscles can be irritable, and that has to do with muscle cells. Anytime you put pressure on them, they would start to respond. So that's the reason we refer to muscle tissues as items that can be irritable. And whenever muscle cells put up energy, they would contract. shorten. And especially with the skeletal muscle cells, and definitely with many of the skeletal muscles, they can be stretched. So they are they have extensibility and somewhat elastic. And we're going to be spending a lot more time with just the skeletal muscle cells here. So to understand these two terms, the myogenesis and morphogenesis. Myogenesis has to do with the development of muscles. And it started with the development of the muscle cells. So the mesoderm are the ones that's going to continually differentiate and then start forming the somites. And now the next item on the somites we deal with would be the myotome. The myotome would continue to become myoblasts. So once the myotome become myoblasts, and the ones that's going to become skeletal muscle cells, they would start fusing. And that's the reason the skeletal muscle fibers would be multinucleated. And definitely with the skeletal muscles, because the muscle cells in each one of these skeletal muscles would have relatively long cylindrical types of cells there. And that's why the muscle cells within a skeletal muscle would be referred to as muscle fibers. So muscle fibers and muscle cells are used interchangeably. Okay. And once they start fusing, then those nuclei are the ones going to start to set up manufacturing the two types of proteins here, myosin and actin. Myosin would be the thick myofilaments, and actin would be the thin myofilaments. And once these two are set up, that muscle would now be able to contract. So when these are set up, that embryo can start moving. The other type of myoblast that's going to be producing cardiac and smooth muscle cells, they do not fuse. So with them, be a single nucleus in the center. Okay. And once they link up as a set of muscles, then they would start linking together with the gap junctions. The cardiac muscle cells are the ones that would be forming a series of cell functions here that stain up. Okay. Then cardiac muscles, even though it hasn't totally formed or even started forming a heart, they can start pumping blood on the 22nd day after fertilization. It started with 2 blood vessels in the median of the body that came together. And those smooth muscles in those 2 blood vessels would start forming the cardiac muscles. Morphogenesis has to do with the organization of the muscle fibers into muscles. And that's around the 5th week. So in other words, by morphogenesis, we are saying that the muscle cells are now completed in forming muscles that eventually we give names to. Things like biceps brachii, so the skeletal muscles would start their contractions by week 7, when the embryo is only 2 centimeters. And eventually, nerves would come to these muscles from the spinal cord. And that's what brings the skeletal muscles under the control of the nervous system. Okay. In a young person around the toddler years, there will be some cells around those cells in the muscle that haven't quite combined with the other cells to form those skeletal muscle fibers. So they would be referred to as satellite cells. But in that young individual, some of those satellite cells can also do some fusion to help those muscles to grow. But once you pass that point, whenever you have an injury, well, at that time, when there is an injury to the muscle, the satellite cells can build up some new muscle cells. But once you get past that stage, no muscle would build up any cells within it. So each muscle is on you would stay to have those same number of muscle cells in them. And throughout the rest of life, every muscle in you cannot build up the muscle cell numbers. They can only lose those muscle cells. Okay. So long cylindrical multinucleated, the nuclei are pushed right up against the cell membranes. So for muscle tissues, they're there to move your skeleton. They can constrict and dilate the organs. Those are the smooth muscles forming the walls of tube-like structures. So they allow to send blood and lymph in those vessels. You have skeletal muscles that can compress your body cavities, especially the abdominal cavity, as well as the internal organs in those cavities. %80 of your body heat is created by muscle contraction. Okay, so constant contraction of muscles would maintain your body temperature. Skeletal muscles are the ones that would be controlling your posture. And eventually we'll come across a series of muscles that would be referred to as postural muscles. So if you're looking at what a muscle will be doing, so going from the outside in, the tendon part of the muscle is the part that doesn't do any contractions, okay? Simply because that is dense, regular connective tissue. You only have fibroblasts in them, and that's it. It's just series of fibers there. And then around a particular muscle, every muscle on your body would be wrapped by epimysium. Epimysium is fibrous material, and especially with the one that is sitting superficial part of the muscle, it would be the one that would have quite a bit of collagen fibers in it. And once you get in there, then you would have a series of muscle cells that would be wrapped by perimysium. And each of these bundles of muscle cells here would be referred to as fascicle. So every one of these ones out here would be something like this, fascicle. And the fascicle will be wrapped around by the perimysium. Perimysium is much thinner than epimysium and will not have as much collagen fibers there. And then once you would come within the fascicle and you start seeing the group of cells that are here wrapping around every cell is endomysium. Endomysium is definitely a lot thinner than the epimysium and perimysium. So for the skeletal muscle cell, okay, that's what this means. Get to know this term sarco. Anytime you see this term sarco, associate that with muscle. So instead of referring to this as plasmalemma, sarcolemma. That's the cell membrane. And then all the nuclei would be pushed right up against it. And sitting outside, maybe some satellite cells. It's not shown here. And then in the within the sarcolemma, you would have mostly those myofibrils. And you also need a lot of mitochondria in there. And then you have the sarcoplasmic reticulum and the terminal cisternae. The terminal cistern would be these two items sandwiching the T-tubule. So those three tube-like structures in here would now be referred to as a triad, also known as transverse tubule. Get to know the T-tubule just by its color. It is material that comes from the sarcolemma, same material as sarcolemma. And there's a reason why you have all these structures here. Has to do with impulses that would be traveling on this cell. And as impulses is going across here, it would continue into the T tubules. As it gets through the T tubules, it's going to cause the material that is in the terminal cisternae, which would be calcium ions in there. It will cause them to release the calcium ions. And when you're looking at these pictures, get to know that the triad is actually where the triad is sitting. If you're looking at this item here, which is called the sarcomere. And you're actually looking at one full sarcomere and two parts of two neighbors. Okay. And within that sarcomere, you have the thick filaments, and then the thin ones. So the blue ones are the actin filaments, and then the thick ones would be the myosin filaments. So can you figure out where the T tubules are right now? If you're looking at that picture.
They're encased by the sarcolemma.
Well, they are, but I'm talking about the triad. Those three items here. The T tubule and the two sarcoplasmic reticula. They will be sitting right over what part of these? Get to know this drawing, okay? Always, the triads would be on every one of these AI junction. And as we continue to go over the sliding filament theory, get to know that. Okay. So for muscle contraction, first we have to get to know the item referred to as the motor unit. Motor unit, I don't test you for the motor unit 1 and motor unit 2. I just need you to know what motor unit means. motor unit has to do with a single motor neuron that's a nerve, which would have the cell body at the anterior gray horn of the spinal cord. By cell body of a neuron, we are saying that that's where the nucleus of these cells would be. So the motor neurons that would be working the skeletal muscle fibers would be here, at the anterior gray horn of the spinal cord. Okay, when we get to the nervous system, we'll be going over what would be on the lateral horn, what would be on the posterior horns. Single motor neuron would have this long process that is called the axon. And that axon would travel all the way to a muscle, and then it's going to start spreading out and forming the neuromuscular junction with a whole series of the muscle cells, okay? Not just one or two as being shown here, okay? It's going to spread and can cover a lot of them. So in the big muscles, a motor neuron can form the neuromuscular junctions to at least 200 muscle fibers. Okay. So in muscles as big as your gluteus maximus, it could be well over 200, close to 300. Okay, just a single motor neuron. Okay. Now, going a little bit further into the neuromuscular junction and what that part of the motor unit is about. At the end of that axon of the motor neuron, you would have a whole series of these axon terminals. And at the end of every one of these terminals, there will be an end bulb right here. And it's going to be linking with the sarcolemma. Now, there would be a synaptic cleft here. And between the two. And in that motor neuron end bulb, you have a whole series of vesicles packed here. And they would be packed with neurotransmitter in granule form. So all these granules are acetylcholine. When the impulse gets to here, then it would cause your standard exocytosis of these, and then the acetylcholine would be sent out. At the sarcolemma, you have those channel proteins, and those channel proteins would have a receptor site for the acetylcholine. Once the acetylcholine get to them, those channels will be open. And once the channel is open, it would start a depolarization. Then that depolarization would be forming this impulse. And the depolarization happens here has to do with all the fluid that is outside of most of the cells in your body. That fluid on the outside would have mostly sodium ions. And once these channels are open, the sodium ions would now enter. And on the other side of the sarcolemma, you normally would have potassium ions. With the sodium ions coming in, then the potassium ions gets forced out. That's what we mean by depolarization. So as that depolarization is happening, like this, then it would follow into the T-tubule. And that would cause sarcolemic cisternae, terminal cisternae, and get them to release calcium ions. So now, can we figure out what happens when you need to move a part of the body? Okay, has to do with you sending an instruction from your brain and get one of these motor neurons to send at least one of these motor neurons to send messages, impulses to depolarize those muscle cells. The next thing that's going to happen your textbook only show you just this part of the sarcomere with with all these terms of it, but what I need you to know is one more items. Okay, I need you to know a little bit more about that actin filament. Okay, so actin filament would be something like this. And it's going to have all these actin molecules. And then each molecule would have a myosin binding site. And you also have the troponin tropomyosin complex that's going to be sitting on them. When a muscle cell is relaxed, that's when the troponin tropomyosin complex would be covering all those myosin binding sites. So the myosin heads would not be reaching for them. But when you send in the impulses to have this cell to constrict, once the sarcoplastic reticulum would be sending out the calcium ions at the terminal cisterna. And because this is sitting at the AI junction, this is where you have the AI junction. Okay. The calcium ions would come to the troponin. Once the calcium ions attach to the troponin, it will cause the troponin tropomyosin complex to slide away and expose all these myosin binding sites on those actin molecules. Now, those myosin heads would now be swinging into these binding sites. And once they swing up, they would grab at it and swing it towards the center of the sarcomere. And that's the process of the sliding filament theory. So also get to know that contraction of every cell in these muscles, every cell, when they are forced to contract, it's always fully contract, totally contract. So, what do we mean by total contraction? When you're looking at the sarcomere, sarcomeres in all of these myofibrils when you look at those parts where you have the two sets of the thin and thick filaments that are not connected. So let's just look at this single sarcomere, okay? So this part here, and then this part here, and this part. Once everything is drawn into the center, do you not consider that this sarcomere would shorten by 1/3 its length. If you add this part to this and that, is that not 1/3 the length of the sarcomere? But now, when the disc, both sides, and all the actin filaments are drawn into the H zone, is this not shortened by 1/3 its length? That's the maximum contraction of all muscle cells. 1/3 the entire length, since both sides are pulled together. Okay. Not talking about not talking about every sarcomere pulling itself apart from the neighbors. Talking about the two ends that is attached to whatever will be drawn in 1/3 its length. So in other words, every muscle on you, the maximum contraction is only 1/3 its length. No muscle can shorten any more than that. And if a muscle starts building up when you do the contraction, that has to do with all these thin filaments stacking up when everything is pulled into the middle. Why is it done like that? Come and look at this picture. If you make a cut right about here at the AI junction, okay, a single myosin can reach its heads out to grab at 6 actin filaments. And each actin filament can be grabbed by the heads of 3 myosin. So that's why it's that perfect. Okay. A little bit about a summary of what I've just gone through. Motor unit. Motor unit consists of a single motor neuron that comes out of the anterior gray horn of the spinal cord, plus all the muscle fibers or muscle cells that is going to be attached to and get them to contract. In a large, coarse, powerful muscle like the gluteus maximus, a given motor neuron may supply 200 muscle fibers. sometimes more than that, somewhere between 200 to 300, depending on how big a person is, and how big those muscles started forming. In the small intrinsic muscles in your hand, or some of the extrinsic muscle that work your eyeball, where you need fine control, one nerve fiber, the motor neuron, would supply only a few muscle fibers. The fewest muscle fibers supplied by a single neuron would be just 6 muscle cells. I think your book has a higher number for that. I don't know where they got that number. Okay. So the muscles you use to move your eyeball around, the muscles you use to blink the eye, and some of the muscles in the middle part of your hand, those are the ones that would have a motor unit working only a couple of dozen at best muscle cells. Okay. At rest, every muscle is in a partial state of contraction. Now we are talking about a muscle, okay, not the muscle cell. Okay. And that condition is often referred to as your muscle tone, tonus of the muscle. And since muscle cells are either fully contracted or relaxed, all or none, okay, no intermediate stages, it follows that a few muscle fibers within a muscle must be fully contracted all the time. To avoid fatigue, different groups of motor units are brought into action by asynchronous depolarization of the somatic motor neurons in the anterior gray horn of the spinal cord at various times, contracting different groups of skeletal muscle fibers within that muscle. Okay. So in other words, within the muscle, and you have different motor units coming to it, and each of those motor units is going to be round and round and round sets of muscles, cells in there to totally contract and totally relax, going over and over and over in a living person. Okay, that's what we mean by tonus of the muscle. Muscle movement is accomplished by bringing into action increasing numbers of motor units in the agonist. Soon you'll be learning this term. Agonist is a prime mover of a muscle for a particular movement. Good example would be your triceps brachii muscle in the back of your arm, back of your humerus. That's the one that extends your forearm. You punch people with your triceps brachii. Okay. It's a prime mover of that extension of your forearm or elbow. And simultaneously reduce the activity of the motor units of muscles that oppose or the antagonists. Muscles that are antagonists of the triceps brachii you actually have three muscles in front of your humerus that will be doing the opposite movement. Biceps brachii, brachialis, brachioradialis, those three muscles, often referred to as the three Bs, they flex the forearm. So that's what we mean. If you want to extend, you are going to bring up the motor units going to your triceps brachii and shut down the ones going to the three muscles sitting in front. And when the maximum effort is required, all the motor units of that muscle will be recruited to contract. Okay. So that's what happens when you start punching someone.
You have experience, Professor?
How's that? So now you know what happens when you do that.
It's good to know for next time.
So now, coming back to the rest of the summary. Okay. Neuromuscular junction is, in other textbooks, you may see these other terms, myo-neuro or myo-neuronal junction. Okay, myo is muscle, neuro, nerve. Okay, so neuromuscular. You have the motor neuron, and then you have that synaptic bulb. That's what this one is often referred to, the end bulb of the axon. And then you have the synaptic vesicle. And then within here, all these around all these synaptic vesicles, you also have quite a bit of mitochondria in here. You need, this is active transfer of material at the cell membrane. Okay, so you need mitochondria there. The synaptic cleft and the material that is within those vesicles, neurotransmitter. In this case, it will be the acetylcholine. So once that happens, once you have enough of the acetylcholine sent out, then you would start the depolarization of sodium and potassium ions. And now it's traveling across the sarcolemma. And then it would follow into the T tubules. And that would cause the cisterna, the end cistern to release calcium ions. Calcium ions come to troponin. Once there, you'll cause the troponin-tropomyosin complex to expose the myosin binding sites on the actin molecules of the actin filament. Once that happens, heads of the myosin swing in, grab at that. Once this is grabbed, you'll swing it towards the middle, and then those heads would Yeah, when they do those, when they do that, that's what we refer to as doing the cross bridges. And then once after swinging, they would go back down and then go back up for the next open myosin site. Okay, so that's why constantly they would be flicking to throw that actin into the H zone of the sarcomere. That all makes sense. So, yeah, I might as well get through this. Your textbook may have a lot more, a few more of these types of muscle fibers. I just need you to note these three, okay? Type 1 fibers, humans largely have type 1 fibers in all their muscles, okay? It's a red, slow twitch, slow tiring fibers. And they have a lot of myoglobin, mitochondria, lots of capillaries to generate ATP through aerobic pathway. Okay, that's the reason it's red, because it's got all these capillaries there, lots of blood there. We have very few muscles that actually would have recognizable fast glycolytic fibers, the type 2X. This is the white, fast twitch, fast tiring fibers. And with these ones, they go after the sugar, and ATP is generated through the anaerobic pathway. The muscles that we have that would be like this would be those muscles that shows up just very light pink. The thin muscles on your eyelid that would do the flexing is the fast twitch, fast tiring fibers that allows you to blink or close your eyes when something comes at your eye. Okay. Mostly we have the type 2A fibers, fast oxidative, intermediate fast twitch fibers. And most likely we have more of this on the gastrocnemius muscle in your calf. Okay, that's the most superficial muscle in the back of your leg, and that's the one used by the fast runners. The muscle deep to it, the soleus muscle, is what we refer to as postural muscle, and that muscle is entirely red. So that's the one that people can use to run long distance, jog, jogging, walking, standing. And that's because it's low twitch, slow tiring muscle. Okay. So those are the 3 sets of fibers. And then getting to know the biomechanics here. And with the biomechanics, we have these 3 classes: leverage system, leverage system. So the first 2 classes is relatively rare on us. Third class is most abundant. So example for the first class here. By the first class, we are going over a structure that has to do with weight that needs to be moved, or the load. The effort has to do with the muscle that is contracting. Fulcrum would be the joint that is responsible for this movement. So in this case, it's the atlanto-occipital joint, talking about the 2 occipital condyles sitting on the superior articular facets of the atlas, C1. And this relatively strong muscle in the back of the neck would have its insertion under the occipital, at the at the bottom of the occipital bone. When it contracts, all this gets hyperextended. Head and neck would be hyperextended. Okay. That's first class. Second class, the material that needs to be moved is the one that's going to be between the joint and the muscle. So here we're looking at gastrocnemius, and it comes with a tendocalcaneus, also known as the it's actually a common tendon of the 3 muscles in the back of your tibia. So both gastrocnemius and soleus are the 2 of those 3 muscles and are the ones responsible for that kind of action. That's one muscle that is relatively very small and This doesn't do that much. But all three of them would have the common tendon right here. So the joint that you have would be the MTP joints across where you have your metatarsals and the proximal phalanges. Okay. And everything between that muscle and the joints would be the item that you need to move. So this is when you lift up on the heel. Dorsiflexion. Okay. Almost every other joint that we go through will be doing this. Third class. Where the muscle is attached to the item that muscle is going to be moving would be between the joint and everything that you need to be moving. So in this case, looking at the biceps brachii muscle, and this is your shoulder joint, where you have your, mostly the one that is responsible for doing this would be the humeral ulna. But since it's together with the radius, so both of them, since they are tied together by the interosseous membrane, all this gets lifted up. Okay. Flexion of the forearm. So, any questions on these? All right then. Let's see you Tuesday.
See you
For some-
Tuesday.
Professor,
Hmm?
I was noticing for the pre-lab, we have to add a little bit more stuff than we have in the past. What do you mean by the origins, insertions, and actions?
Okay. uh, sometime this weekend, I'm going to change the due dates on a few things here. So check into the e-text where you have Chapter 11. Go to table 11.8 and 11.9. There, it's going to be giving you the list of the origins and insertions of the muscles there. Okay. And I don't mind if you send that in on the 29th. And on the 29th, I will go over the answer key on those. All right. On Tuesday, I'll be explaining origin insertions, tendons, everything. So page 7B on the lecture outline would have all those terms. Okay. I'll be going through all that, and then I'll start going into a series of muscles on Tuesday. Okay.
Okay, thanks, Professor.
Thank you.
Can I ask you something after everyone is left?
Is there another question?
Professor, I have one question. We have an essay due on Wednesday. Since it seems like our groups are kind of in flux because people are thinking about dropping and things like that, is there a possibility we could push that back?
Push what back?
The essay?
When is that due?
According to the assignment, I think it says June 30th.
Really? Oh. Let's see. When do I put my?
It's in the bottom of the all the way at the bottom.
I know.
You were right above it. Go all the way down.
I know. Let's move it to July 6th.
Thank you very much, Professor.
Okay.
Thank you so much, Professor.
Yeah, I'll change that.
Thank you.
Yeah, it needs to be done before the second exam. So, yeah. So go with July 6th, okay? All right. So I'll start closing. Good night.
Good night,
Good night.
Professor. Have a good one.